Habitat. In ecology, habitat refers to the array of resources, physical and biotic factors that are present in an area, such as to support the survival and reproduction of a particular species. A species' habitat can be seen as the physical manifestation of its ecological niche. Thus, habitat is a species-specific term, fundamentally different from concepts such as environment or vegetation assemblages, for which the term habitat type is more appropriate. 2. This coral reef in the Phoenix Islands protected area provides habitat for numerous marine species. Few creatures make the ice shelves of Antarctica their habitat, but water beneath the ice can provide habitat for multiple species. Animals such as penguins have adapted to live in very cold conditions. 1. Ibex in an alpine habitat. The physical factors may include, for example, soil, moisture, range of temperature, and light intensity. Biotic factors include the availability of food and the presence or absence of predators. Every species has particular habitat requirements. Habitat generalist species are able to thrive in a wide array of environmental conditions, while habitat specialist species require a very limited set of factors to survive. The habitat of a species is not necessarily found in a geographical area. It can be the interior of a stem, a rotten log, a rock, or a clump of moss. A parasitic organism has as its habitat the body of its host, part of the host's body, such as the digestive tract, or a single cell within the host's body. 3. Habitat types are environmental categorizations of different environments based on the characteristics of a given geographical area, particularly vegetation and climate. 2. Thus, habitat types do not refer to a single species but to multiple species living in the same area. For example, terrestrial habitat types include forest, steppe, grassland, semi-arid or desert. Freshwater habitat types include marshes, streams, rivers, lakes, and ponds. Marine habitat types include salt marshes, the coast, the intertidal zone, estuaries, reefs, bays, the open sea, the seabed, deep water, and submarine vents. Habitat types may change over time. Causes of change may include a violent event, such as the eruption of a volcano, an earthquake, a tsunami, a wildfire, or a change in oceanic currents, or change may occur more gradually over millennia with alterations in the climate, as ice sheets and glaciers advance and retreat, and as different weather patterns bring changes of precipitation and solar radiation. Other changes come as a direct result of human activities, such as deforestation, the plowing of ancient grasslands, the diversion and damming of rivers, the draining of marshland, and the dredging of the seabed. The introduction of alien species can have a devastating effect on native wildlife, through increased predation, through competition for resources, or through the introduction of pests and diseases to which the indigenous species have no immunity. Definition and etymology. The word habitat has been in use since about 1755 and derives from the Latin habitar, to inhabit, from haber, to hold. Habitat can be defined as the natural environment of an organism, the type of place in which it is natural for it to live and grow. 4.5. It is similar in meaning to a biotope, an area of uniform environmental conditions associated with a particular community of plants and animals. 6. Environmental factors. The chief environmental factors affecting the distribution of living organisms are temperature, humidity, climate, soil and light intensity, and the presence or absence of all the requirements that the organism needs to sustain it. Generally speaking, animal communities are reliant on specific types of plant communities. 7. Some plants and animals have habitat requirements which are met in a wide range of locations. The small white butterfly Pieris rapi, for example, is found on all the continents of the world apart from Antarctica. Its larvae feed on a wide range of brassicas and various other plant species, and it thrives in any open location with diverse plant associations. 8. The large blue butterfly Phangaris aurean is much more specific in its requirements. It is found only in chalk grassland areas, its larvae feed on thymus species, and because of complex life cycle requirements it inhabits only areas in which Myrmica ants live. 9. Disturbance is important in the creation of biodiverse habitat types. In the absence of disturbance, a climax vegetation cover develops that prevents the establishment of other species. Wildflower meadows are sometimes created by conservationists, but most of the flowering plants used are either annuals or biennials and disappear after a few years in the absence of patches of bare ground on which their seedlings can grow. 10. Lightning strikes and toppled trees in tropical forests allow species richness to be maintained as pioneering species move in to fill the gaps created. 11. Similarly, coastal habitat types can become dominated by kelp until the seabed is disturbed by a storm and the algae swept away, or shifting sediment exposes new areas for colonization. Another cause of disturbance is when an area may be overwhelmed by an invasive introduced species which is not kept under control by natural enemies in its new habitat. 12. Types. Terrestrial. Rich rainforest habitat in Dominica. Terrestrial habitat types include forests, grasslands, wetlands, and deserts. Within these broad biomes are more specific habitat types with varying climate types, temperature regimes, soils, altitudes, and vegetation. Many of these habitat types grade into each other and each one has its own typical communities of plants and animals. A habitat type may suit a particular species well, but its presence or absence at any particular location depends to some extent on chance, on its dispersal abilities and its efficiency as a colonizer. 13. Arid. Desert scene in Egypt. Arid habitats are those where there is little available water. The most extreme arid habitats are deserts. Desert animals have a variety of adaptations to survive the dry conditions. Some frogs live in deserts, creating moist habitat types underground and hibernating while conditions are adverse. Couch's spadefoot toad, Scaphiopas cutchi, emerges from its burrow when a downpour occurs and lays its eggs in the transient pools that form. The tadpoles develop with great rapidity, sometimes in as little as 9 days, undergo metamorphosis, and feed voraciously before digging a burrow of their own. 14. List of arid habitat types. Desert. Fog desert. Polar desert. Steppe. Savanna. Wetland and riparian. Other organisms cope with the drying up of their aqueous habitat in other ways. Vernal pools are ephemeral ponds that form in the rainy season and dry up afterwards. They have their specially adapted characteristic flora, mainly consisting of annuals, the seeds of which survive the drought, but also some uniquely adapted perennials. 15. Animals adapted to these extreme habitat types also exist. Fairy shrimps can lay winter eggs, which are resistant to desiccation, sometimes being blown about with the dust, ending up in new depressions in the ground. These can survive in a dormant state for as long as 15 years. 16. Some killifish behave in a similar way. Their eggs hatch and the juvenile fish grow with great rapidity when the conditions are right, but the whole population of fish may end up as eggs in diapause in the dried up mud that was once a pond. 17. Examples of wetland and riparian habitat types. Bog. Marsh. Fen. Flooded grasslands and savannas floodplain. Shrub swamp. Swamp. Vernal pool. Wet meadow forest. Examples of forest habitat types. Boreal forest. Cloud forest. Peat swamp forest. Temperate coniferous forest, temperate deciduous forest, temperate rain forest. Thorn forest. Tropical dry forest. Tropical moist forest, tropical rain forest, woodland. Tropical moist forest, tropical rain forest, woodland. Freshwater. Wetland habitat types in Borneo. Freshwater habitat types include rivers, streams, lakes, ponds, marshes and bogs. 18. They can be divided into running waters, rivers, streams, and standing waters, lakes, ponds, marshes, bogs. 19. Although some organisms are found across most of these habitat types, the majority have more specific requirements. The water velocity, its temperature and oxygen saturation are important factors, but in river systems, there are fast and slow sections, pools, bayous and backwaters which provide a range of habitat types. Similarly, aquatic plants can be floating, semi-submerged, submerged or grow in permanently or temporarily saturated soils besides bodies of water. Marginal plants provide important habitat for both invertebrates and vertebrates, and submerged plants provide oxygenation of the water, absorb nutrients and play a part in the reduction of pollution. 20. List of freshwater habitat types. Rivers, ponds, lakes. Marine. Marine habitats include brackish water, estuaries, bays, the open sea, the intertidal zone, the seabed, reefs and deep, shallow water zones. 18. Further variations include rock pools, sandbanks, mudflats, brackish lagoons, sandy and pebbly beaches, and seagrass beds, all supporting their own flora and fauna. The benthic zone or seabed provides a home for both static organisms, anchored to the substrate, and for a large range of organisms crawling on or burrowing into the surface. Some creatures float among the waves on the surface of the water, or raft on floating debris, others swim at a range of depths, including organisms in the demersal zone close to the seabed, and myriads of organisms drift with the currents and form the plankton. 21. List of marine habitat types. Abyssal plain, aphotic zone, benthic zone, cold sea. Coral reef. Demersal zone, estuary. Hydrothermal vent, intertidal zone, kelp forest. Littoral zone, oceanic trench, photic zone. Seagrass meadow, mangrove swamp, seamount. Tide pool. Urban. Many animals and plants have taken up residence in urban environments. They tend to be adaptable generalists and use the town's features to make their homes. Rats and mice have followed man around the globe. Pigeons, peregrines, sparrows, swallows and house martins use the buildings for nesting. Bats use roof space for roosting. Foxes visit the garbage bins and squirrels. Coyotes, raccoons and skunks roam the streets. About 2,000 coyotes are thought to live in and around Chicago. 22. A survey of dwelling houses in northern European cities in the 20th century found about 175 species of invertebrate inside them, including 53 species of beetle, 21 flies, 13 butterflies and moths, 13 mites, 9 lice, 7 bees, 5 wasps, 5 cockroaches, 5 spiders, 4 ants and a number of other groups. 23. In warmer climates, termites are serious pests in the urban habitat. 183 species are known to affect buildings and 83 species cause serious structural damage. 24. Microhabitat types. The leaves of an Alnus nepalensis tree provide a microhabitat for species like the leaf beetle Alocaphora indica. A microhabitat is the small-scale physical requirements of a particular organism or population. Every habitat includes large numbers of microhabitat types with subtly different exposure to light, humidity, temperature, air movement, and other factors. The lichens that grow on the north face of a boulder are different from those that grow on the south face, from those on the level top, and those that grow on the ground nearby. The lichens growing in the grooves and on the raised surfaces are different from those growing on the veins of quartz. Lurking among these miniature forests are the microfauna, species of invertebrate, each with its own specific habitat requirements. 25. There are numerous different microhabitat types in a wood: coniferous forest, broadleafed forest, open woodland, scattered trees, woodland verges, clearings, and glades, tree trunk, branch, twig, bud, leaf, flower, and fruit, rough bark, smooth bark, damaged bark, rotten wood, hollow, groove, and hole, canopy, shrub layer, plant layer, leaf litter, and soil, buttress root, stump, fallen log, stem base, grass tussock, fungus, fern, and moss. 26. The greater the structural diversity in the wood, the greater the number of microhabitat types that will be present. A range of tree species with individual specimens of varying sizes and ages, and a range of features such as streams, level areas, slopes, tracks, clearings, and felled areas will provide suitable conditions for an enormous number of biodiverse plants and animals. For example, in Britain it has been estimated that various types of rotting wood are home to over 1,700 species of invertebrate. 26. For a parasitic organism, its habitat is the particular part of the outside or inside of its host on or in which it is adapted to live. The life cycle of some parasites involves several different host species, as well as free-living life stages, sometimes within vastly different microhabitat types. 27. One such organism is the trematode flatworm, Microphallus turgidus, present in brackish water marshes in the southeastern United States. Its first intermediate host is a snail and the second, a glass shrimp. The final host is the waterfowl or mammal that consumes the shrimp. 28. Extreme habitat types. An Antarctic rock split apart to show endolithic lifeforms showing as a green layer a few millimeters thick. Although the vast majority of life on Earth lives in mesophilic moderate environments, a few organisms, most of them microbes, have managed to colonize extreme environments that are unsuitable for more complex life forms. There are bacteria, for example, living in Lake Whillans, half a mile below the ice of Antarctica. In the absence of sunlight, they must rely on organic material from elsewhere, perhaps decaying matter from glacier meltwater or minerals from the underlying rock. 29. Other bacteria can be found in abundance in the Mariana Trench, the deepest place in the ocean and on Earth. Marine snow drifts down from the surface layers of the sea and accumulates in this undersea valley, providing nourishment for an extensive community of bacteria. 30. Other microbes live in environments lacking in oxygen, and are dependent on chemical reactions other than photosynthesis. Boreholes drilled 300 meters 1,000 feet into the rocky seabed have found microbial communities apparently based on the products of reactions between water and the constituents of rocks. These communities have not been studied much, but may be an important part of the global carbon cycle. 31. Rock in mines 2 miles deep also harbor microbes. These live on minute traces of hydrogen produced in slow oxidizing reactions inside the rock. These metabolic reactions allow life to exist in places with no oxygen or light, an environment that had previously been thought to be devoid of life. 32. 33. The intertidal zone and the photic zone in the oceans are relatively familiar habitat types. However, the vast bulk of the ocean is inhospitable to air-breathing humans, with scuba divers limited to the upper 50 meters 160 feet or so. 34. The lower limit for photosynthesis is 100 to 200 meters 330 to 660 feet, and below that depth the prevailing conditions include total darkness, high pressure, little oxygen, in some places, scarce food resources and extreme cold. This habitat is very challenging to research, and as well as being little studied, it is vast, with 79% of the Earth's biosphere being at depths greater than 1,000 meters 3,300 feet. 35. With no plant life, the animals in this zone are either detritivores, reliant on food drifting down from surface layers, or they are predators, feeding on each other. Some organisms are pelagic, swimming or drifting in mid-ocean, while others are benthic, living on or near the seabed. Their growth rates and metabolisms tend to be slow, their eyes may be very large to detect what little illumination there is, or they may be blind and rely on other sensory inputs. A number of deep-sea creatures are bioluminescent. This serves a variety of functions including predation, protection and social recognition. 35. In general, the bodies of animals living at great depths are adapted to high-pressure environments by having pressure-resistant biomolecules and small organic molecules present in their cells known as piezolites, which give the proteins the flexibility they need. There are also unsaturated fats in their membranes which prevent them from solidifying at low temperatures. 36. Dense mass of white crabs at a hydrothermal vent, with stocked barnacles on right. Hydrothermal vents were first discovered in the ocean depths in 1977. 37. They result from seawater becoming heated after seeping through cracks to places where hot magma is close to the seabed. The underwater hot springs may gush forth at temperatures of over 340 degrees Celsius, 640 degrees Fahrenheit, and support unique communities of organisms in their immediate vicinity. 37. The basis for this teeming life is chemosynthesis, a process by which microbes convert such substances as hydrogen sulfide or ammonia into organic molecules. 38. These bacteria and archaea are the primary producers in these ecosystems and support a diverse array of life. About 350 species of organism, dominated by mollusks, polychaete worms and crustaceans, had been discovered around hydrothermal vents by the end of the 20th century, most of them being new to science and endemic to these habitat types. 39. Besides providing locomotion opportunities for winged animals and a conduit for the dispersal of pollen grains, spores and seeds, the atmosphere can be considered to be a habitat type in its own right. There are metabolically active microbes present that actively reproduce and spend their whole existence airborne, with hundreds of thousands of individual organisms estimated to be present in a cubic meter of air. The airborne microbial community may be as diverse as that found in soil or other terrestrial environments. However, these organisms are not evenly distributed, their densities varying spatially with altitude and environmental conditions. Aerobiology has not been studied much, but there is evidence of nitrogen fixation in clouds, and less clear evidence of carbon cycling, both facilitated by microbial activity. 40. There are other examples of extreme habitat types where specially adapted life forms exist. Tar pits teeming with microbial life. 41. Naturally occurring crude oil pools inhabited by the larvae of the petroleum fly. 42. Hot springs where the temperature may be as high as 71 degrees Celsius, 160 degrees Fahrenheit, and cyanobacteria create microbial mats. 43. Cold seeps where the methane and hydrogen sulfide issue from the ocean floor and support microbes and higher animals such as mussels which form symbiotic associations with these anaerobic organisms. 44. Salt pans that harbor salt tolerant. 40. There are other examples of extreme habitat types where specially adapted life forms exist. Tar pits teeming with microbial life. 41. Naturally occurring crude oil pools inhabited by the larvae of the petroleum fly. 42. Hot springs where the temperature may be as high as 71 degrees Celsius, 160 degrees Fahrenheit, and cyanobacteria create microbial mats. 43. Cold seeps where the methane and hydrogen sulfide issue from the ocean floor and support microbes and higher animals such as mussels which form symbiotic associations with. There are other examples of extreme habitat types where specially adapted life forms exist. Tar pits teeming with microbial life. 41. Naturally occurring crude oil pools inhabited by the larvae of the petroleum fly. 42. Hot springs where the temperature may be as high as 71 degrees Celsius, 160 degrees Fahrenheit, and cyanobacteria create microbial mats. 43. Cold seeps where the methane and hydrogen sulfide issue from the ocean floor and support microbes and higher animals such as mussels which form symbiotic associations with these anaerobic organisms. 44. Salt pans that harbor salt-tolerant bacteria, archaea and also fungi such as the black yeast Hordeia wernicki and Basidiomycete Wallamia ichthyophaga. 45. 46. Ice sheets in Antarctica which support fungi Thelebulus spp. 45. Glacial ice with a variety of bacteria and fungi. 47. And snowfields on which algae grow. 48. Habitat change. 25 years after the devastating eruption at Mount St. Helens, United States, pioneer species have moved in. Whether from natural processes or the activities of man, landscapes and their associated habitat types change over time. There are the slow geomorphological changes associated with the geologic processes that cause tectonic uplift and subsidence, and the more rapid changes associated with earthquakes, landslides, storms, flooding, wildfires, coastal erosion, deforestation and changes in land use. 49. Then there are the changes in habitat types brought on by alterations in farming practices, tourism, pollution, fragmentation and climate change. 50. Loss of habitat is the single greatest threat to any species. If an island on which an endemic organism lives becomes uninhabitable for some reason, the species will become extinct. Any type of habitat surrounded by a different habitat is in a similar situation to an island. If a forest is divided into parts by logging, with strips of cleared land separating woodland blocks, and the distances between the remaining fragments exceeds the distance an individual animal is able to travel, that species becomes especially vulnerable. Small populations generally lack genetic diversity and may be threatened by increased predation, increased competition, disease and unexpected catastrophe. 50. At the edge of each forest fragment, increased light encourages secondary growth of fast-growing species and old-growth trees are more vulnerable to logging as access is improved. The birds that nest in their crevices, the epiphytes that hang from their branches and the invertebrates in the leaf litter are all adversely affected and biodiversity is reduced. 50. Habitat fragmentation can be ameliorated to some extent by the provision of wildlife corridors connecting the fragments. These can be a river, ditch, strip of trees, hedgerow or even an underpass to a highway. Without the corridors, seeds cannot disperse and animals, especially small ones, cannot travel through the hostile territory, putting populations at greater risk of local extinction. 51. Habitat disturbance can have long-lasting effects on the environment. Bromus tectorum is a vigorous grass from Europe which has been introduced to the United States where it has become invasive. It is highly adapted to fire, producing large amounts of flammable detritus and increasing the frequency and intensity of wildfires. It is highly adapted to fire, producing large amounts of flammable detritus and increasing the frequency and intensity of wildfires. In areas where it has become established, it has altered the local fire regimen to such an extent that native plants cannot survive the frequent fires, allowing it to become even more dominant. 52. A marine example is when sea urchin populations explode in coastal waters and destroy all the macroalgae present. What was previously a kelp forest becomes an urchin barren that may last for years, and this can have a profound effect on the food chain. Removal of the sea urchins, by disease for example, can result in the seaweed returning, with an overabundance of fast-growing kelp. 53. Fragmentation. Predicted fragmentation and destruction of great ape habitat in Central Africa, from the GLOBIO, 54, and GRASP projects in 2002. Areas shown in black and red delineate areas of severe and moderate habitat loss, respectively. Deforestation in Europe. France is the most deforested country in Europe, with only 15% of the native vegetation remaining. Deforestation in Bolivia, 2016. Habitat fragmentation describes the emergence of discontinuities fragmentation in an organism's preferred environment, habitat causing population fragmentation and ecosystem decay. 55. Causes of habitat fragmentation include geological processes that slowly alter the layout of the physical environment. 56. Suspected of being one of the major causes of speciation. 56. And human activity such as land conversion, which can alter the environment much faster and causes the extinction of many species. More specifically, habitat fragmentation is a process by which large and contiguous habitats get divided into smaller, isolated patches of habitats. 57-58. Destruction. Map of the world's biodiversity hotspots, all of which are heavily threatened by habitat loss and degradation. Habitat destruction, also termed habitat loss and habitat reduction, occurs when a natural habitat is no longer able to support its native species. The organisms once living there have either moved elsewhere or are dead, leading to a decrease in biodiversity and species numbers. 59-60. Habitat destruction is in fact the leading cause of biodiversity loss and species extinction worldwide. 61. Humans contribute to habitat destruction through the use of natural resources, agriculture, industrial production and urbanization, urban sprawl. Other activities include mining, logging and trawling. Environmental factors can contribute to habitat destruction more indirectly. Geological processes, climate change, 60. Introduction of invasive species, ecosystem nutrient depletion, water and noise pollution are some examples. Loss of habitat can be preceded by an initial habitat fragmentation. Fragmentation and loss of habitat have become one of the most important topics of research in ecology as they are major threats to the survival of endangered species. 62. Habitat protection. The protection of habitat types is a necessary step in the maintenance of biodiversity because if habitat destruction occurs, the animals and plants reliant on that habitat suffer. Many countries have enacted legislation to protect their wildlife. This may take the form of the setting up of national parks, forest reserves and wildlife reserves, or it may restrict the activities of humans with the objective of benefiting wildlife. The laws may be designed to protect a particular species or group of species, or the legislation may prohibit such activities as the collecting of bird eggs, the hunting of animals or the removal of plants. A general law on the protection of habitat types may be more difficult to implement than a site-specific requirement. A concept introduced in the United States in 1973 involves protecting the critical habitat of endangered species, and a similar concept has been incorporated into some Australian legislation. 63. International treaties may be necessary for such objectives as the setting up of marine reserves. Another international agreement, the Convention on the Conservation of Migratory Species of Wild Animals, protects animals that migrate across the globe and need protection in more than one country. 64. Even where legislation protects the environment, a lack of enforcement often prevents effective protection. However, the protection of habitat types needs to take into account the needs of the local residents for food, fuel and other resources. Faced with hunger and destitution, a farmer is likely to plow up a level patch of ground despite it being the last suitable habitat for an endangered species such as the San Quintin kangaroo rat, and even kill the animal as a pest. 65. In the interests of ecotourism, it is desirable that local communities are educated on the uniqueness of their flora and fauna. 66. Monotypic habitat. A monotypic habitat type is a concept sometimes used in conservation biology, in which a single species of animal or plant is the only species of its type to be found in a specific habitat and forms a monoculture. Even though it might seem such a habitat type is impoverished in biodiversity as compared with polytypic habitat types, this is not necessarily the case. Monocultures of the exotic plant hydrilla support a similarly rich fauna of invertebrates as a more varied habitat. 67. The monotypic habitat occurs in both botanical and zoological contexts. Some invasive species may create monocultural stands that prevent other species from growing there. A dominant colonization can occur from retardant chemicals exuded, nutrient monopolization, or from lack of natural controls, such as herbivores or climate, that keep them in balance with their native habitat types. The yellow star thistle, Centaurea solstitialis, is a botanical monotypic habitat example of this, currently dominating over 15 million acres, 61,000 square kilometers, in California alone. 68. The non-native freshwater zebra mussel, Dryzena polymorpha, that colonizes areas of the Great Lakes and the Mississippi River watershed, is a zoological monotypic habitat example. The predators or parasites that control it in its home range in Russia are absent. Density dependence. In population ecology, density-dependent processes occur when population growth rates are regulated by the density of a population. 1. This article will focus on density dependence in the context of macroparasite life cycles. Positive density dependence. Positive density density-dependent facilitation, or the Allais effect, describes a situation in which population growth is facilitated by increased population density. Examples. In dioecious, separate-sex, obligatory parasites, mated female worms are required to complete a transmission cycle. At low parasite densities, the probability of a female worm encountering a male worm and forming a mating pair can become so low that reproduction is restricted due to single-sex infections. At higher parasite densities, the probability of mating pairs forming and successful reproduction increases. This has been observed in the population dynamics of schistosomes. 2. Positive density dependence processes occur in macroparasite life cycles that rely on vectors with a subarrial armature, such as Anopheles or Culex mosquitoes. For Wuchereria bancrofti, a filarial nematode, well-developed subarrial armatures in vectors can damage ingested microfilariae and impede the development of infective L3 larvae. At low microfilariae densities, most microfilariae can be ruptured by teeth, preventing successful development of infective L3 larvae. As more larvae are ingested, the ones that become entangled in the teeth may protect the remaining larvae, which are then left undamaged during ingestion. 3. Positive density dependence processes may also occur in macroparasite infections that lead to immunosuppression. Onchocerca infection promotes immunosuppressive processes within the human host that suppress immunity against incoming infective L3 larvae. This suppression of anti-parasite immunity causes parasite establishment rates to increase with higher parasite burden. 4. Negative density dependence. Negative density dependence, or density-dependent restriction, describes a situation in which population growth is curtailed by crowding, predators and competition. In cell biology, it describes the reduction in cell division. When a cell population reaches a certain density, the amount of required growth factors and nutrients available to each cell becomes insufficient to allow continued cell growth. This is also true for other organisms because an increased density means an increase in intraspecific competition. Greater competition means an individual has a decreased contribution to the next generation, i.e. offspring. Density-dependent mortality can be overcompensating, undercompensating or exactly compensating. There also exists density-independent inhibition, where other factors such as weather or environmental conditions and disturbances may affect a population's carrying capacity. An example of a density-dependent variable is crowding and competition. Examples. Density-dependent fecundity. Density-dependent fecundity exists, where the birth rate falls as competition increases. In the context of gastrointestinal nematodes, the weight of female Ascaris lumbricoides and its rates of egg production decreases, host infection intensity increases. Thus, the per capita contribution of each worm to transmission decreases as a function of infection intensity. 5. Parasite-induced vector mortality. Parasite-induced vector mortality is a form of negative density dependence. The onchocerciasis life cycle involves transmission via a black fly vector. In this life cycle, the life expectancy of the black fly vector decreases as the worm load ingested by the vector increases. Because ovolvulus microfilariae require at least 7 days to mature into infective L3 larvae in the black fly, the worm load is restricted to levels that allow the black fly to survive for long enough to pass infective L3 larvae onto humans. 6. In macroparasite life cycles. Density dependence processes, red, in filariasis life cycle. In macroparasite life cycles, density-dependent processes can influence parasite fecundity, survival, and establishment. Density-dependent processes can act across multiple points of the macroparasite life cycle. For filarial worms, density-dependent processes can act at the host-vector interface or within the host-vector life cycle stages. At the host-vector interface, density dependence may influence the input of L3 larvae into the host skin and the ingestion of microfilariae by the vector. Within the life cycle stages taking place in the vector, density dependence may influence the development of L3 larvae in vectors and vector life expectancy. Within the life cycle stages taking place in the host, density dependence may influence the development of microfilariae and host life expectancy. 7. In reality, combinations of negative restriction and positive facilitation density-dependent processes occur in the life cycles of parasites. However, the extent to which one process predominates over the other vary widely according to the parasite, vector, and host involved. This is illustrated by the W. Bancrofti life cycle. In Culex mosquitoes, which lack a well-developed subarrial armature, restriction processes predominate. Thus, the number of L3 larvae per mosquito declines as the number of ingested microfilariae increases. Conversely, in Aedes and Anopheles mosquitoes, which have well-developed subarrial armatures, facilitation processes predominate. Consequently, the number of L3 larvae per mosquito increases as the number of ingested microfilariae increases. 3. Implications for parasite persistence and control. Negative density-dependent restriction processes contribute to the resilience of macroparasite populations. At high parasite populations, restriction processes tend to restrict population growth rates and contribute to the stability of these populations. Interventions that lead to a reduction in parasite populations will cause a relaxation of density-dependent restrictions, increasing per capita rates of reproduction or survival, thereby contributing to population persistence and resilience. 7. Contrariwise, positive density-dependent or facilitation processes make elimination of a parasite population more likely. Facilitation processes cause the reproductive success of the parasite to decrease with lower worm burden. Thus, control measures that reduce parasite burden will automatically reduce per capita reproductive success and increase the likelihood of elimination when facilitation processes predominate. 8. Extinction threshold. The extinction threshold refers to minimum parasite density level for the parasite to persist in a population. Interventions that reduce parasite density to a level below this threshold will ultimately lead to the extinction of that parasite in that population. Facilitation processes increase the extinction threshold, making it easier to achieve using parasite control interventions. Conversely, restriction processes complicates control measures by decreasing the extinction threshold. 8. Implications for parasite distribution. Anderson and Gordon, 1982, propose that the distribution of macroparasites in a host population is regulated by a combination of positive and negative density-dependent processes. In overdispersed distributions, a small proportion of hosts harbor most of the parasite population. Positive density-dependent processes contribute to overdispersion of parasite populations, whereas negative density-dependent processes contribute to underdispersion of parasite populations. As mean parasite burden increases, negative density-dependent processes become more prominent and the distribution of the parasite population tends to become less overdispersed. 9. Consequently, interventions that lead to a reduction in parasite burden will tend to cause the parasite distribution to become overdispersed. For instance, time series data for onchocerciasis infection demonstrates that 10 years of vector control lead to reduced parasite burden with a more overdispersed distribution. 10. See also. Populations grow at geometric or exponential rates in the presence of unlimited resources. Geometric populations grow through pulsed reproduction, e.g., the annual reproduction of deer, which have a constrained mating and reproduction season. Exponential populations grow continuously, with reproduction occurring at any time, such as among humans. All populations begin exponential growth in favorable environments and at low population densities. Because of this, exponential growth may apply to populations establishing new environments, during transient, favorable conditions, and by populations with low initial population density. However, geometrical or exponential growth cannot continue indefinitely. In nature, population growth must eventually slow, and population size ceases to increase. As resources are depleted, population growth rate slows and eventually stops. This is known as logistic growth. The population size at which growth stops is generally called the carrying capacity K, which is the number of individuals of a particular population that the environment can support. At carrying capacity, because population size is approximately constant, birth rates must equal death rates, and population growth is zero. Populations cannot grow without limit. No population can increase without limitation. Instead, populations in natural ecosystems increase or decrease in response to the changes in the factors that restrict growth. Many factors influence population densities and growth, and these factors may lead to oscillations in population size over time. It is also often difficult to determine the exact factor limiting growth. Many different factors may combine to produce unexpected results. One organism that experiences rapid oscillations in population density in response to growth-limiting factors is the lemming. Lemmings are small rodents that live in the high Arctic tundra of Greenland and in other Arctic environments across the world. One species, the collared lemming Dicrostonyx groenlandicus, is a chubby-looking rodent living in the Arctic of North America and Greenland. It is food for a number of vertebrate predators, including the stoat, a short-tailed weasel, the Arctic fox, the snowy owl, and the long-tailed skua, a seabird. Because of the simplicity of this system, lemming population dynamics make an excellent case study for examining the factors regulating population growth. Gilg et al. 2003 studied this system in the Karup Valley of northeast Greenland. The lemming population increased and decreased in a regular 4-year cycle during the study period, 1988-2002, Figure 1. The number of lemmings increased to as many as 10 per hectare. Gilg et al. 2003 found that the single most important factor limiting lemming population size was the predation pressure affecting those populations. The owl, fox, and skua switched to lemming predation as the lemming numbers increased, preventing rapid population growth. As the lemmings provided the stoat with additional food, their reproductive success increased, allowing an increased stoat population. Stoat population expansion eventually overran lemming population growth, and the lemming population collapsed, soon followed by a collapse in the stoat population, and the cycle repeated itself. Density-dependent limitation. Limitations to population growth are either density-dependent or density-independent. Density-dependent factors include disease, competition, and predation. Density-dependent factors can have either a positive or a negative correlation to population size. With a positive relationship, these limiting factors increase with the size of the population and limit growth as population size increases. With a negative relationship, population growth is limited at low densities and becomes less limited as it grows. This page has been archived and is no longer updated. Citation. Figure 2. Mortality of juvenile red deer from 0 to 24 months of age, hollow circles, and mortality of adults between 5 to 10 years old, Cluttenbrock et al. 2002. Copyright 2011 Nature Education, all rights reserved. Density-dependent factors may influence the size of the population by changes in reproduction or survival. Wouters and Lenz 1995 studied how food availability and density combine to limit red squirrel reproduction rates. The red squirrel, Sciurus vulgaris, is a small rodent inhabiting forests in Europe and Asia. They studied squirrels in both coniferous and deciduous woodlands and investigated how limitations in food resulted in limitations in reproduction as population densities increased. They found that when squirrel densities were high, territoriality relegated some females to poor quality territory, which in turn reduced their reproductive success. When squirrel densities were low, no females occupied the low quality territory. Thus, it was not all individuals suffering from reduced ability to reproduce, e.g., fecundity, due to the density increase. Instead, a greater proportion of the population was living in poor quality habitat, while those still living in good habitat continued to have success. This in turn led to a decrease in per capita birth rate. A limitation in population growth is a function of population density. Density-dependent factors may also affect population mortality and migration. Cluttenbrock et al. 2002 found these density-dependent controls in a population of red deer, Cervicolophus, in the Scottish Highlands. Both juvenile and adult mortality was significantly affected by population density, with juvenile mortality more strongly influenced than adult mortality, Figure 2. Furthermore, they found that these differences were stronger among males than females, so that increasing population density caused a shift in the sex ratio of females to males. This effect was enhanced by decreased male immigration and increased male emigration. Thus, density-dependent controls on population growth not only increased with increasing density, but also differentially affected males and females within the population. Density-independent limitation. Factors that decrease population growth can be defined as environmental stress, including limitations in food, predation, and other density-dependent factors, Sibley and Hone 2002. However, many sources of environmental stress affect population growth, irrespective of the density of the population. Density-independent factors, such as environmental stressors and catastrophe, are not influenced by population density change. While the previously mentioned density-dependent factors are often biotic, density-independent factors are often abiotic. These density-independent factors include food or nutrient limitation, pollutants in the environment, and climate extremes, including seasonal cycles such as monsoons. In addition, catastrophic factors can also impact population growth, such as fires and hurricanes. The quality of nutrients, e.g., food quality, amount of particular plant nutrients, in an environment affects the ability of an organism to survive, grow, and reproduce. The lower the quality of the nutrients, the higher the environmental stress. In the freshwater Laurentian Great Lakes, particularly in Lake Erie, the factor limiting algal growth was found to be phosphorus. David Schindler and his colleagues at the Experimental Lakes Area, Ontario, Canada demonstrated that phosphorus was the growth-limiting factor in temperate North American lakes using whole lake treatment and controls, Schindler 1974. This work encouraged the passage of the Great Lakes Water Quality Agreement of 1972, GLWQA 1972. A reduction in phosphorus load from municipal sources was predicted to lead to a corresponding reduction in the total algal biomass and harmful cyanobacterial, blue-green algae, blooms, McGuckin 2000, Figure 3. As annual phosphorus loads decreased in the mid-1980s, Dolan 1993, there was some indication that Lake Erie was improving in terms of decreased total phytoplankton, photosynthetic algae and cyanobacteria, biomass, Makarovich 1993. Further improvement continued until the mid-1990s, until an introduced species, the zebra mussel, began altering the internal phosphorus dynamics of the lake by mineralization, excretion, of digested algae, Figure 3, Conroy et al. Outline, keywords. Basic. Advanced. Aging and its demographic measurement allay effects. An introduction to population growth, climate change and avian population ecology in Europe. Density and dispersion. McGuckin, W. Lake. Pollutants also contribute to environmental stress, limiting the growth rates of populations. Although each species has specific tolerances for environmental toxins, amphibians in general are particularly susceptible to pollutants in the environment. For example, pesticides and other endocrine-disrupting toxins can strongly control the growth of amphibians, Blaustein et al. 2003. These chemicals are used to control agricultural pests but also run into freshwater streams and ponds where amphibians live and breed. They affect the amphibians both with direct increases in mortality and indirect limitation in growth, development, and reduction in fecundity. Rohr et al. 2003 found, among many other examples, that these compounds affect salamander embryo survival in affected ponds, increased deformities, and delayed development and growth, lengthening their vulnerability to predators by remaining small size for longer periods. These effects limit population growth irrespective of the size of the amphibian population and are not limited to pesticides but also include pH and thermal pollution, herbicides, fungicides, heavy metal contaminations, etc. Environmental catastrophes such as fires, earthquakes, volcanoes and floods can strongly affect population growth rates via direct mortality and habitat destruction. A large-scale natural catastrophe occurred in 2005 when Hurricane Katrina impacted the coastal regions of the Gulf of Mexico in the southern United States. Katrina altered habitat for coastal vegetation by depositing more than 5 centimeters of sediment over the entire coastal wetland zone. In these areas, substantial improvement in the quality of wetlands for plant growth occurred after many years of wetland loss due to control of the Mississippi River flow, Turner et al. 2006. At the same time, however, almost 100 kilometers of wetland was destroyed and converted to open sea, completely eliminating wetland vegetation, Day et al. 2007. More recently, the Gulf oil spill in 2010 has again impacted the coastal wetland vegetation. Though human-derived, this large-scale environmental disaster will have long-term impacts on the population growth of not only vegetation but all organisms in the wetlands and nearshore regions of the Gulf of Mexico. Metapopulation. A metapopulation consists of a group of spatially separated populations of the same species which interact at some level. The term metapopulation was coined by Richard Levins in 1969 to describe a model of population dynamics of insect pests in agricultural fields, but the idea has been most broadly applied to species in naturally or artificially fragmented habitats. In Levins' own words, it consists of a population of populations. 1. Metapopulations are important in fisheries. The local population 1 serves as a source for hybridization with surrounding subspecies populations 1a, 1b, and 1c. The populations are normally spatially separated and independent, but spatial overlap between them during breeding times allows for gene flow between the populations. A metapopulation is generally considered to consist of several distinct populations together with areas of suitable habitat which are currently unoccupied. In classical metapopulation theory, each population cycles in relative independence of the other populations and eventually goes extinct as a consequence of demographic stochasticity, fluctuations in population size due to random demographic events. The smaller the population, the more chances of inbreeding depression and prone to extinction. Although individual populations have finite lifespans, the metapopulation as a whole is often stable because immigrants from one population, which may, for example, be experiencing a population boom, are likely to recolonize habitat which has been left open by the extinction of another population. They may also emigrate to a small population and rescue that population from extinction, called the rescue effect. Such a rescue effect in metapopulation. A metapopulation, in Levin's own words, they may also serves as a source. A metapopulation is generally considered to consist of several distinct populations together with areas of suitable habitat which are currently unoccupied. In classical metapopulation theory, each population cycles in relative independence of the other populations and eventually goes extinct as a consequence of demographic stochasticity, fluctuations in population size due to random demographic events. The smaller the population, the more chances of inbreeding depression and prone to extinction. Although individual populations have finite lifespans, the metapopulation as a whole is often stable because immigrants from one population, which may, for example, be experiencing a population boom, are likely to recolonize habitat which has been left open by the extinction of another population. They may also emigrate to a small population and rescue that population from extinction, called the rescue effect. Such a rescue effect may occur because declining populations leave niche opportunities open to the rescuers. The development of metapopulation theory, in conjunction with the development of source sink dynamics, emphasize the importance of connectivity between seemingly isolated populations. Although no single population may be able to guarantee the long-term survival of a given species, the combined effect of many populations may be able to do this. Metapopulation theory was first developed for terrestrial ecosystems, and subsequently applied to the marine realm. Metapopulation theory was first developed for terrestrial ecosystems, and subsequently applied to the marine realm. 2. In fisheries science, the term, subpopulation, is equivalent to the metapopulation science term, local population. Most marine examples are provided by relatively sedentary species occupying discrete patches of habitat, with both local recruitment and recruitment from other local populations in a larger metapopulation. Kritzer and Sale have argued against strict application of the metapopulation definitional criteria that extinction risks to local populations must be non-negligible. 232. Finnish biologist Ilpa Hansky of the University of Helsinki was an important contributor to metapopulation theory. Predation and oscillations. The first experiments with predation and spatial heterogeneity were conducted by G. F. Gause in the 1930s, based on the Lotka-Volterra equation, which was formulated in the mid-1920s, but no further application had been conducted. 3. The Lotka-Volterra equation suggested that the relationship between predators and their prey would result in population oscillations over time based in the initial densities of predator and prey. Gause's early experiments to prove the predicted oscillations of this theory failed because the predator-prey interactions were not influenced by immigration. However, once immigration was introduced, the population cycles accurately depicted the oscillations predicted by the Lotka-Volterra equation, with the peaks in prey abundance shifted slightly to the left of the peaks of the predator densities. Huffaker's experiments expanded on those of Gause by examining how both the factors of migration and spatial heterogeneity lead to predator-prey oscillations. Huffaker's experiments on predator-prey interactions, 1958. In order to study predation and population oscillations, Huffaker used mite species, one being the predator and the other being the prey. 4. He set up a controlled experiment using oranges, which the prey fed on, as the spatially structured habitat in which the predator and prey would interact. 5. At first, Huffaker experienced difficulties similar to those of Gause in creating a stable predator-prey interaction. By using oranges only, the prey species quickly became extinct, followed consequently with predator extinction. However, he discovered that by modifying the spatial structure of the habitat, he could manipulate the population dynamics and allow the overall survival rate for both species to increase. He did this by altering the distance between the prey and oranges, their food, establishing barriers to predator movement, and creating corridors for the prey to disperse. 3. These changes resulted in increased habitat patches and in turn provided more areas for the prey to seek temporary protection. When the prey would become extinct locally at one habitat patch, they were able to re-establish by migrating to new patches before being attacked by predators. This habitat spatial structure of patches allowed for coexistence between the predator and prey species and promoted a stable population oscillation model. 6. Although the term metapopulation had not yet been coined, the environmental factors of spatial heterogeneity and habitat patchiness would later describe the conditions of a metapopulation relating to how groups of spatially separated populations of species interact with one another. Huffaker's experiment is significant because it showed how metapopulations can directly affect the predator-prey interactions and in turn influence population dynamics. 7. The Levin's model. Levin's original model applied to a metapopulation distributed over many patches of suitable habitat with significantly less interaction between patches than within a patch. Population dynamics within a patch were simplified to the point where only presence and absence were considered. Each patch in his model is either populated or not. Let n be the fraction of patches occupied at a given time. During a time dt, each occupied patch can become unoccupied with an extinction probability at. Additionally, 1 minus n of the patches are unoccupied. Assuming a constant rate c of propagule generation from each of the n occupied patches, during a time dt, each unoccupied patch can become occupied with a colonization probability cn dt. Accordingly, the time rate of change of occupied patches, dn dt, is. This equation is mathematically equivalent to the logistic model, with a carrying capacity k given by and growth rate r. At equilibrium, therefore, some fraction of the species' habitat will always be unoccupied. Stochasticity in metapopulations. Huffaker's 4 studies of spatial structure and species interactions are an example of early experimentation in metapopulation dynamics. Since the experiments of Huffaker, 4, and Levins, 1 models have been created which integrate stochastic factors. These models have shown that the combination of environmental variability, stochasticity, and relatively small migration rates cause indefinite or unpredictable persistence. However, Huffaker's experiment almost guaranteed infinite persistence because of the controlled immigration variable. Stochastic patch occupancy models, SPOMs. One major drawback of the Levins model is that it is deterministic, whereas the fundamental metapopulation processes are stochastic. Metapopulations are particularly useful when discussing species in disturbed habitats, and the viability of their populations, i.e., how likely they are to become extinct in a given time interval. The Levins model cannot address this issue. A simple way to extend the Levins model to incorporate space and stochastic considerations is by using the contact process. Simple modifications to this model can also incorporate for patch dynamics. At a given percolation threshold, habitat fragmentation effects take place in these configurations, predicting more drastic extinction thresholds. 8. For conservation biology purposes, metapopulation models must include a. The finite nature of metapopulations, how many patches are suitable for habitat, and b. The probabilistic nature of extinction and colonization. Also, in order to apply these models, the extinctions and colonizations of the patches must be asynchronous. Microhabitat patches, MHPs, and bacterial metapopulations. E. Coli metapopulation on a chip. Combining nanotechnology with landscape ecology, synthetic habitat landscapes have been fabricated on a chip by building a collection of bacterial mini-habitats with nanoscale channels, providing them with nutrients for habitat renewal, and connecting them by corridors in different topological arrangements, generating a spatial mosaic of patches of opportunity distributed in time. This can be used for landscape experiments by studying the bacteria metapopulations on the chip, for example their evolutionary ecology. 9. Life history evolution. Metapopulation models have been used to explain life history evolution, such as the ecological stability of amphibian metamorphosis in small vernal ponds. Alternative ecological strategies have evolved. For example, some salamanders forego metamorphosis and sexually mature as aquatic neotens. The seasonal duration of wetlands and the migratory range of the species determines which ponds are connected and if they form a metapopulation. The duration of the life history stages of amphibians relative to the duration of the vernal pool before it dries up regulates the ecological development of metapopulations connecting aquatic patches to terrestrial patches. Theoretical ecology. Theoretical ecology is the scientific discipline devoted to the study of ecological systems using theoretical methods such as simple conceptual models, mathematical models, computational simulations, and advanced data analysis. Effective models improve understanding of the natural world by revealing how the dynamics of species populations are often based on fundamental biological conditions and processes. Further, the field aims to unify a diverse range of empirical observations by assuming that common, mechanistic processes generate observable phenomena across species and ecological environments. Based on biologically realistic assumptions, theoretical ecologists are able to uncover novel, non-intuitive insights about natural processes. Theoretical results are often verified by empirical and observational studies, revealing the power of theoretical methods in both predicting and understanding the noisy, diverse biological world. Mathematical models developed in theoretical ecology predict complex food webs can be less stable than simpler webs. 1, 75-77, 2, 64. Life on Earth, flow of energy and entropy. The field is broad and includes foundations in applied mathematics, computer science, biology, statistical physics, genetics, chemistry, evolution, and conservation biology. Theoretical ecology aims to explain a diverse range of phenomena in the life sciences, such as population growth and dynamics, fisheries, competition, evolutionary theory, epidemiology, animal behavior and group dynamics, food webs, ecosystems, spatial ecology, and the effects of climate change. Theoretical ecology has further benefited from the advent of fast computing power, allowing the analysis and visualization of large-scale computational simulations of ecological phenomena. Importantly, these modern tools provide quantitative predictions about the effects of human-induced environmental change on a diverse variety of ecological phenomena, such as species invasions, climate change, the effect of fishing and hunting on food network stability, and the global carbon cycle. Modeling approaches. As in most other sciences, mathematical models form the foundation of modern ecological theory. Phenomenological models distill the functional and distributional shapes from observed patterns in the data, or researchers decide on functions and distribution that are flexible enough to match the patterns they or others, field or experimental ecologists, have found in the field or through experimentation. 3. Mechanistic models model the underlying processes directly, with functions and distributions that are based on theoretical reasoning about ecological processes of interest. 3. Ecological models can be deterministic or stochastic. 3. Deterministic models always evolve in the same way from a given starting point. 4. They represent the average, expected behavior of a system, but lack random variation. Many system dynamics models are deterministic. Stochastic models allow for the direct modeling of the random perturbations that underlie real-world ecological systems. Markov chain models are stochastic. Species can be modeled in continuous or discrete time. 5. Continuous time is modeled using differential equations. Discrete time is modeled using difference equations. These model ecological processes that can be described as occurring over discrete time steps. Matrix algebra is often used to investigate the evolution of age-structured or stage-structured populations. The Leslie matrix, for example, mathematically represents the discrete time change of an age-structured population. 6, 7, 8. Models are often used to describe real ecological reproduction processes of single or multiple species. These can be modeled using stochastic branching processes. Examples are the dynamics of interacting populations, predation, competition, and mutualism, which, depending on the species of interest, may best be modeled over either continuous or discrete time. Other examples of such models may be found in the field of mathematical epidemiology, where the dynamic relationships that are to be modeled are host-pathogen interactions. 5. Bifurcation diagram of the logistic map. Bifurcation theory is used to illustrate how small changes in parameter values can give rise to dramatically different long-run outcomes, a mathematical fact that may be used to explain drastic ecological differences that come about in qualitatively very similar systems. 9. Logistic maps are polynomial mappings, and are often cited as providing archetypal examples of how chaotic behavior can arise from very simple nonlinear dynamical equations. The maps were popularized in a seminal 1976 paper by the theoretical ecologist Robert May. 10. The difference equation is intended to capture the two effects of reproduction and starvation. In 1930, R.A. Fisher published his classic The Genetical Theory of Natural Selection, which introduced the idea that frequency-dependent fitness brings a strategic aspect to evolution, where the payoffs to a particular organism, arising from the interplay of all of the relevant organisms, are the number of this organism's viable offspring. 11. In 1961, Richard Lewontin applied game theory to evolutionary biology in his Evolution and the Theory of Games. 12. Followed closely by John Maynard Smith, who in his seminal 1972 paper, Game Theory and the Evolution of Fighting, 13, defined the concept of the evolutionarily stable strategy. Because ecological systems are typically nonlinear, they often cannot be solved analytically, and in order to obtain sensible results, nonlinear, stochastic and computational techniques must be used. One class of computational models that is becoming increasingly popular are the agent-based models. These models can simulate the actions and interactions of multiple, heterogeneous, organisms where more traditional, analytical techniques are inadequate. Applied theoretical ecology yields results which are used in the real world. For example, optimal harvesting theory draws on optimization techniques developed in economics, computer science and operations research, and is widely used in fisheries. 14. Population ecology. Population ecology is a subfield of ecology that deals with the dynamics of species populations and how these populations interact with the environment. 15. It is the study of how the population sizes of species living together in groups change over time and space, and was one of the first aspects of ecology to be studied and modeled mathematically. Exponential growth. The most basic way of modeling population dynamics is to assume that the rate of growth of a population depends only upon the population size at that time and the per capita growth rate of the organism. In other words, if the number of individuals in a population at a time t is nt, then the rate of population growth is given by where r is the per capita growth rate, or the intrinsic growth rate of the organism. It can also be described as r equals bd, where b and d are the per capita time invariant birth and death rates, respectively. This first-order linear differential equation can be solved to yield the solution. A trajectory known as Malthusian growth, after Thomas Malthus, who first described its dynamics in 1798. A population experiencing Malthusian growth follows an exponential curve, where n0 is the initial population size. The population grows when r greater than 0, and declines when r less than 0. The model is most applicable in cases where a few organisms have begun a colony and are rapidly growing without any limitations or restrictions impeding their growth, e.g. bacteria inoculated in rich media. Logistic growth. The exponential growth model makes a number of assumptions, many of which often do not hold. For example, many factors affect the intrinsic growth rate and is often not time invariant. A simple modification of the exponential growth is to assume that the intrinsic growth rate varies with population size. This is reasonable. The larger the population size, the fewer resources available, which can result in a lower birth rate and higher death rate. Hence, we can replace the time invariant r with rt equals bant d plus c asterisk nt, where n c are constants that modulate birth and death rates in a population-dependent manner, e.g. intraspecific competition. Both a and circa will depend on other environmental factors which, we can for now, assume to be constant in this approximated model. The differential equation is now, 16. This can be rewritten as, 16. Where r equals bd and k equals bd, a plus c. The biological significance of k becomes apparent when stabilities of the equilibria of the system are considered. The constant k is the carrying capacity of the population. The equilibria of the system are n equals 0 and n equals k. If the system is linearized, it can be seen that n equals 0 is an unstable equilibrium while k is a stable equilibrium. 16. Structured population growth. Another assumption of the exponential growth model is that all individuals within a population are identical and have the same probabilities of surviving and of reproducing. This is not a valid assumption for species with complex life histories. The exponential growth model can be modified to account for this, by tracking the number of individuals in different age classes, e.g. 1, 2, and 3-year-olds, or different stage classes, juveniles, subadults, and adults separately, and allowing individuals in each group to have their own survival and reproduction rates. The general form of this model is where n is a vector of the number of individuals in each class at time t and L is a matrix that contains the survival probability and fecundity for each class. The matrix L is referred to as the Leslie matrix for age-structured models, and is the Lefkovich matrix for stage-structured models. 17. If parameter values in L are estimated from demographic data on a specific population, a structured model can then be used to predict whether this population is expected to grow or decline in the long term, and what the expected age distribution within the population will be. This has been done for a number of species including loggerhead sea turtles and right whales. 18-19. Community ecology. An ecological community is a group of trophically similar, sympatric species that actually or potentially compete in a local area for the same or similar resources. 20. Interactions between these species form the first steps in analyzing more complex dynamics of ecosystems. These interactions shape the distribution and dynamics of species. Of these interactions, predation is one of the most widespread population activities. 21. Taken in its most general sense, predation comprises predator-prey, host-pathogen, and host-parasitoid interactions. Lotka-Volterra model of cheetah-baboon interactions. Starting with 80 baboons, green, and 40 cheetahs, this graph shows how the model predicts the two species numbers will progress over time. Predator-prey interaction. Predator-prey interactions exhibit natural oscillations in the populations of both predator and the prey. 21. In 1925, the American mathematician Alfred J. Lotka developed simple equations for predator-prey interactions in his book on biomathematics. 22. The following year, the Italian mathematician Vito Volterra made a statistical analysis of fish catches in the Adriatic. 23. And independently developed the same equations. 24. It is one of the earliest and most recognized ecological models, known as the Lotka-Volterra model. Where n is the prey and p is the predator population sizes, r is the rate for prey growth, taken to be exponential in the absence of any predators, alpha is the prey mortality rate for per capita predation, also called attack rate, c is the efficiency of conversion from prey to predator, and d is the exponential death rate for predators in the absence of any prey. Volterra originally used the model to explain fluctuations in fish and shark populations after fishing was curtailed during the First World War. However, the equations have subsequently been applied more generally. 25. Other examples of these models include the Lotka-Volterra model of the snowshoe hare and Canadian lynx in North America. 26. Any infectious disease modeling such as the recent outbreak of SARS. 27. And biological control of California red scale by the introduction of its parasitoid, Aphidus melanus. 28. A credible, simple alternative to the Lotka-Volterra predator-prey model and their common prey-dependent generalizations is the ratio-dependent or Arditi-Ginzburg model. 29. The two are the extremes of the spectrum of predator interference models. According to the authors of the alternative view, the data show that true interactions in nature are so far from the Lotka-Volterra extreme on the interference spectrum that the model can simply be discounted as wrong. They are much closer to the ratio-dependent extreme, so if a simple model is needed one can use the Arditi-Ginzburg model as the first approximation. 30. Host-pathogen interaction. The second interaction, that of host and pathogen, differs from predator-prey interactions in that pathogens are much smaller, have much faster generation times, and require a host to reproduce. Therefore, only the host population is tracked in host-pathogen models. Compartmental models that categorize host population into groups such as susceptible, infected, and recovered (SIR) are commonly used. 31. Host-parasitoid interaction. The third interaction, that of host and parasitoid, can be analyzed by the Nicholson-Bailey model, which differs from Lotka-Volterra and SIR models in that it is discrete in time. This model, like that of Lotka-Volterra, tracks both populations explicitly. Typically, in its general form, it states, where f ent, part describes the probability of infection typically, Poisson distribution lambda is the per capita growth rate of hosts in the absence of parasitoids, and c is the conversion efficiency, as in the Lotka-Volterra model. 21. Competition and mutualism. In studies of the populations of two species, the Lotka-Volterra system of equations has been extensively used to describe dynamics of behavior between two species, N1 and N2. Examples include relations between D. discoiderum and E. coli, 32, as well as theoretical analysis of the behavior of the system. 33. The R coefficients give a base growth rate to each species, while K coefficients correspond to the carrying capacity. What can really change the dynamics of a system, however, are the alpha terms. These describe the nature of the relationship between the two species. When alpha 12 is negative, it means that N2 has a negative effect on N1, by competing with it, preying on it, or any number of other possibilities. When alpha 12 is positive, however, it means that N2 has a positive effect on N1, through some kind of mutualistic interaction between the two. When both alpha 12 and alpha 21 are negative, the relationship is described as competitive. In this case, each species detracts from the other, potentially over competition for scarce resources. When both alpha 12 and alpha 21 are positive, the relationship becomes one of mutualism. In this case, each species provides a benefit to the other, such that the presence of one aids the population growth of the other. See competitive Lotka-Volterra equations for further extensions of this model. Neutral theory. Unified neutral theory is a hypothesis proposed by Stephen P. Hubble in 2001. 20. The hypothesis aims to explain the diversity and relative abundance of species in ecological communities, although like other neutral theories in ecology, Hubble's hypothesis assumes that the differences between members of an ecological community of trophically similar species are neutral, or irrelevant to their success. Neutrality means that at a given trophic level in a food web, species are equivalent in birth rates, death rates, dispersal rates and speciation rates, when measured on a per capita basis. 34. This implies that biodiversity arises at random, as each species follows a random walk. 35. This can be considered a null hypothesis to niche theory. The hypothesis has sparked controversy, and some authors consider it a more complex version of other null models that fit the data better. Under unified neutral theory, complex ecological interactions are permitted among individuals of an ecological community, such